Hello, I'm Adrian, and in this video I would like to give you a short tutorial to the Bacillus subtilis Genome Atlas, or BSGatlas for short. The BSGatlas is a resource that integrates multiple databases into one single resource. These existing databases that we integrate provide a wide variety of different annotation types. And by integrating them in one spot, you, the user, have access to functional annotations, non-coding RNA and structured RNA annotations, but also full transcripts in one single resource. The BSGatlas is also available as a browser, in which by interactive means you can investigate the annotation, but also add additional data that you might be interested to inspect in relationship to the BSGatlas. We also computed a list of guide RNAs for which we predict potential off-targets and on-targets that you might be interested in using when designing your own CRISPR experiment. If you're interested in reading about how the BSGatlas was generated, please have a look at our recently published manuscript in the Microbial Genomics journal. This tutorial has three main parts. At first, we have a brief look at the BSGatlas website. Afterwards, we have a look at the genome browser and how to interact with it and how to read the off-target assessment reports presented there and how to potentially add additional data here in a showcase a public RNA-seq experiment. Finally, but not least, we'll have a brief look at the offline use cases that the BSGatlas can have, which will be incredibly helpful, especially in high throughput analysis scenarios. Now, first, the website as a short overview. If we go to RTHDK and select the BSGatlas, we present it with a section that has essentially three main components. The website provides a navigation bar here on the left, in which we can select different type of gene records, for instance, enzyme classifications or functional annotations from the gene ontology, here in the middle shown, which has biological processes, and by clicking on one of the processes, we see which genes are associated with it. Other relevant records are the enzyme classification systems. We have from SubtiWiki a species-specific category system. We have also from an RFM-scan a quite comprehensive list of RNA structures. And from the CAC database, we know possible pathways in which predominantly coding genes are annotated. The BSGatlas annotates various gene types, which are potentially organized in operons. For each of these cases, we provide record pages. Finally, when either clicking in the records on one of the gene names or using the search field to search for a gene of interest, we are presented with a detailed list of information regarding each gene. On the one hand, we provide coordinates of the annotation, but also with respect to external databases, what additional meta information is available. Now let's have a look at the website. Let's click on the link. We are presented with the BSGatlas. If we, for instance, select a gene of interest from, let's say, the arginine biosynthesis pathway, we have a web page that contains all the details relating to, in this case, this glutaminase, GLSA. On the bottom of the page, so let's scroll down, we provide a short insight into the genomic neighborhood of GLSA. Here we have a small browser window in which we have on the top gene annotations. In blue we have coding genes with coloration and indicating the strand of location. Underneath we have operons and transcriptional information, and depending on presence of potentially alternative transcription start sites, we might have also isoforms that are shorter than the entire operon. So let's zoom out a bit. Right here, for instance, we have an example of an operon that has transcription start size that give rise to short isoforms. Over here, we see a gray annotation. The gray coloration indicates a riboswitch. If you would like to know more, we can also click directly on the annotation to go to the corresponding detailed page. In this case, this specific riboswitch is a T-box. Given that short introduction to the website, let's turn to the Genome browser. The Genome browser provides a more powerful view on the BSGatlas with a much wider variety of content in it. Thus, in this part of the tutorial, we'll give a short overview of what kind of content there is, how you can customize working with it, and what kind of built-in tools might be helpful in your research. Afterwards, we'll present briefly the off-target reports that we computed and show you how you might be able to integrate additional RNA-seq data information in the browser. The Genome browser has, at first glance, a similar structure to the browser that we showed on the website. If we start from top to bottom, we have on the top gene annotations with blue indicating coding genes. Underneath we have isoform annotations, operons, untranslated regions, and transcription start and termination sites. However, even more information that is shown underneath. Besides our potential guide RNAs, we have here the comprehensive signal from a massive tiling array study. This specific study in question has been published in Science by Nicholas et al. It contains transcription signals for over 100 different conditions. Thus, given such a large number of conditions, it becomes increasingly difficult to navigate them. Also here, we provide in the Genome browser a solution. Let's have a look at it. How do we get to the Genome browser? On the website, there's here in the navigation bar an entry for the browser. Here we select a mirror that is geographically closer to our locations. In my case, that is Europe. Now we are on the UCC Genome Browser Gateway. This gateway provides us annotation for a wide number of different species, from human all the way down to mouse, rat, and additional. Because we arrived here over the BSGatlas website, all the way on the top, we have an entry we can select in this gateway to go to the browser for the BSGatlas. If we simply press on go, now we're presented with a view on the Genome browser as just shown. We have here the coding genes, transcripts, operons, all the way down to the tiling array and the guide RNAs. When we further scroll down on this web page, we can see a control panel that allows us to, in detail, select the kind of tracks we would like to see in the browser. We can also click on each track to further adjust a wide variety of different settings. Now I clicked on the tiling array for the forward strand. Here we have a variety of different settings we can adjust. More importantly, here on the bottom, we see which of those over 100 environmental conditions are shown. We can also select a specific subset of conditions that we are interested in looking at, for instance, this growth condition, and for beautiful contrast, this glucose metabolic change condition. If we now click here on the top on submit, we have now reduced which specific conditions are shown. Similarly, if we want to have the exact same conditions shown for the reverse strand, we would need to also change the settings here. So we click on the track and select the exact conditions we're interested in looking at. And then click on Submit. Let's have a look at what additional information can be shown in this browser. So let's go to the control panel. Currently shown are the gene annotations, transcripts, operons, and transcription start/termination sites of the BSGatlas. We can hide individual tracks by selecting in these drop-down menus the hide options. Let's do that for some options to clean out a bit the browser. Now we're left with the gene annotation and the guide RNAs. Additional tracks we could include would be the transcriptional units. These are annotations that were used as an intermediate steps to derive the transcripts. We can also look at the gene annotations as they were in the individual databases that were used when generating the BSGatlas. The browser also has the option to show enzyme restriction sites which are potentially of interest when designing an experiment. Similar to the gene annotations, we can also look at the details of annotations with respect to transcription start and termination sites from various resources. Each track has different visibility options besides just plainly hiding it. On the one hand here, the genes are currently shown in a packed form. That would be in contrast to the full version where each gene would have a separate row per entry. The pack option is especially helpful when zooming out of the browser would result in a too large number of gene annotations. Besides packing, one can squish annotations, which results in a very narrow band of annotation tracks, or even more densely packed. The dense visibility indicates regions that contain annotations, although overlapping annotations are not separated. Personally, I prefer the packed visibility for the genes and squish for the transcripts and operons. Another customization option is to drag and drop on the browser. On the one hand, we can go left and right, but we could also drag individual tracks up or down to change their position within the browser. A helpful tool would also be to press and hold the Shift key and highlight a certain region in the browser. So here we have a small pop-up window which allows us to add a colored highlight to a region, or we can also select a region to which we zoom in. Now if we zoom in closer on a gene, we finally see potential guide RNAs that are shown below here. In order to understand how these guide RNAs were predicted, let's have a look at how CRISPR functions. In this figure we have an illustration how Cas9 binds in complex with a guide RNA to DNA in order to introduce a specific modification. The level of how well the Cas9 complex binds to DNA is predominantly affected by complementarity of the guide to the genome. In contrast to on-target regions in which a guide has perfect complementarity, off-targets are bound by the guide with mismatches. The inclination of a guide Cas9 complex to bind to both on and off-target regions can be assessed with an energy-specific model as described by Alkan et al. We use this energy model to estimate the specificity for each possible guide RNA in the Bacillus genome. With that in mind, let's have a look at the report. Let's click on one of those guides. Here we see a report for this specific guide sequence and this AGT PAM. There is only one region to which this guide binds with perfect complementarity. Thus, the potential on-target of this guide is at this region. Potential genes that are affected by this on-target region are listed here. And the first 3 columns indicate how well the CRISPR-Cas9 guide complex binds to that region and how specific it is to this region. The specificity of a guide is estimated from the list of all off-target scores, both potential on and off targets, including off-target regions of up to 6 mismatches. Due to the large number of such regions, we here show only off targets for up to 4 mismatches. With this short overview into the guide RNA track, let's have a look at other features of the Genome browser. The Genome browser has a variety of many tools it provides to a user. Let's highlight a few that might be most helpful for your research. One is that in this view option, a user can choose the DNA option to view the exact DNA sequence of the region that is shown in the Genome browser. If we copy this specific part of this region and return back to the browser, we can use the LAT tool, the basic local alignment tool, to also search for sequences that are similar to what we just copied. In this case, only a single region has a match. If we go to the browser, we see now an additional track that highlights the exact region to which the DNA we just copied aligns. An additional helpful view is the PDF export. By clicking on the PDF export, we can get a figure of the Genome browser, which we can use in reports or manuscripts. Now let's have a look at how we might be able to add additional information, for instance from an RNA-seq experiment, to the browser. To showcase such an integration, I use publicly available RNA-seq data, in this case from Nagler et al. I downloaded the data from the sequence read archive and mapped them against the Bacillus genome. If you're interested in learning how to conduct a quality control step and map reads against the genome, and finally retrieve a coverage signal from an RNA-seq experiment, I would recommend that you have a look at the training material of the Galaxy project. In combination, the computational steps will provide a file that provides for genome coordinates the coverage signal. These coverage signals can be directly loaded into the Genome browser. Let's do that. Back in the Genome browser, we have here a button that allows us to add additional track information. Here I only have to choose the file that I just prepared from the gene mapping and click Submit. After the browser has processed my upload, I'm presented with a table of samples I've just integrated into the Genome browser. Here I have the forward and reverse strand for 2 exemplary samples. I can get additional settings for each sample by clicking on them. Here I can change the name or the color with which the track will be shown in the Genome browser. In the Genome browser, I've now the forward and reverse strands shown for both cases. If we revisit now the upload form that we just used to integrate the data, we're presented here on top with a list of different file formats that can be readily integrated in the Genome browser. If you wish to learn more on how to configure the tracks that were integrated, we can click here and we're being presented with a list of various options. That brings us to the final part of this tutorial. how can we use the BSGatlas in an offline setting? So on the BSGatlas website, we have a download page in which you can download the annotation in an Excel file format. However, you can also choose the GFF file format. This file is so far remarkable as it contains the entire annotation in a single file, but also preserves the relationship between annotations. Thus, you can use this file on your local machine with, for instance, the integrative genomics viewer, which also allows you to load transcriptome covered files, similar to what was just shown earlier. However, this file can also be used for more advanced analyses, such as differential genome expression analysis. To understand the GFF file, it might help to think like a computer sequentially in the steps of transcription. So a start site gives rise to a transcript, and the RNA molecule contains the structures such as a riboswitch and the termination structure. The coding sequences are derived from genomic locations then on the DNA. On an operon organization level, it can occur that multiple genes are located within the same operon. With these relationships in mind, I hope you can use the GFF file in your research. In short summary, the BSGatlas contains a comprehensive list of functional annotations, non-coding, structured RNA annotations, entire transcripts, but also provides access via an interactive browser, and yet moreover has a list of potential guide RNAs and off-targets. I would like to thank my collaborators for making the BSGatlas possible, and thank you Innovation Fund Denmark for supporting this work. If you have more questions concerning the BSGatlas, please do not hesitate to write me an email to adrian@rth.dk. Thank you and see you next time.